Which of the following does not kill endospores? A. Autoclaving. B. Incineration. C. Hot air sterilization. D. Pasteurization. E. All of the above kill endospores. Well, A. Autoclaving, that entails that the temperature would have to be 121 degrees Celsius or 15 psi for 15 minutes. So that can kill an endospore. So it cannot be A. So we're going to cross that one out. Incineration deals with direct fire, and in that method, endospores can be killed. Hot air sterilization involves 170 degrees Celsius for 2 hours. So this method would kill endospores. What we have left is pasteurization, and all of the above will kill endospores. Well, pasteurization entails hot temperature, short time pasteurization, which has the temperature of 72 degrees Celsius for 15 seconds. Well, that's not hot enough to kill endospores. And so the answer would be D. Yeah, we can cross out E. Next one. Which of the following is most effective for sterilizing mattresses and plastic petri dishes? A. Well, chlorine can be used for drinking water, pools, or sewages. B. Ethylene oxide or ethanol kills all microbes and endospores, but requires lengthy exposure periods of greater hours, increased hours. C. Glutaraldehyde. Well, it's used to disinfect hospital instruments, but not practical. It's not a practical sterilant. D. Autoclaving is a method of sterilization in the healthcare environment. All right. And E. Non-ionizing radiation. Well, radiation is not very penetrating. So out of all of these, which one of the following is most effective for sterilizing mattresses and plastic petri dishes? Well, the answer is B. Ethylene oxide or ethanol, because it kills all microbes and endospores. Which of these disinfectants does not act by disrupting the plasma membrane? Let's find the ones that do. Phenolics injures the lipid-containing plasma membrane. Phenol is a significant antibacterial chemical, so we can cross these two out. Quats. It affects the plasma membrane, thus changing the cell's permeability. So we can cross that out. What we are left are with halogens and biguanides. Well, biguanides has a mode of action that affects bacterial cell membrane. You can cross E out. What we are left with is D, halogens. So that is the answer. We have an example of a halogen, which is iodide, which affects the protein synthesis, not the plasma membrane. Which of the following cannot be used to sterilize a heat labile solution stored in a plastic container? A. Gamma radiation. B. Ethylene oxide. C. Supercritical fluids. D. Autoclaving. And E. Short wavelength radiation. We need to know what is heat labile solution. And heat labile solution is a solution that is capable of changing or being destroyed when subjected to heat. Which of these options contain heat? Well, it's D. D contains heat. If heat is applied, then the solution will be destroyed or it will change, and that's not what is wanted. So the answer is D. Which of the following is used to control microbial growth in foods? Organic acids, which are additives that the body can metabolize and are food safe. Alcohols, which are topical antiseptics or disinfectants, which cannot be put in foods, not food safe. Aldehydes, which is a topical disinfectant, which is not safe. And heavy metals. Well, silver nanoparticles can be infused in plastic food containers to keep food fresh, or all of the above. Well, it's to keep food fresh, but you cannot put it in food. But we do have organic acids, additives such as sodium benzoate or sorbic acid. So the answer is A. Additives, organic acids. This table here shows the comparison of 4 disinfectants against Salmonella cholerae suis. We have G for growth and NG for no growth. Now, which disinfectant is the most effective? Well, we want to know which one shows that there is no growth. And we see here disinfectant A shows the most no growth. So the answer is A. Disinfectant A. Now, which disinfectant or disinfectants is or are bactericidal? Which ones kill bacteria? Well, we see no growth for A. We see growth for B, so that's not it. We see no growth for C, and we see a no growth for D. Now we're looking for A, C, and D. A, C, and D. B. So the answer is B. Which of the following is not a characteristic of quaternary ammonium compounds? Well, it's easy to find what are the characteristics. Eventually, it'll lead us to the one that is not. So we need to find the ones that are characteristic. We have the first one, bactericidal, against gram-positive bacteria. That is true. Sporicidal, we'll leave that one. Ambicidal, that's true. Fungicidal, that's true. And kills enveloped viruses, that is true. What we are left with is sporicidal. The answer is B. A classmate is trying to determine how a disinfectant might kill cells. So he wants to know the disinfectant might kill cells. You observe that when he spills the disinfectant in your reduced litmus milk, the litmus turns blue. You suggest to your classmate the following: that inhibit cell wall synthesis, oxidize molecules, inhibit protein synthesis, denatures protein, or he takes his work away from yours. So you want to know if how the disinfectant might kill the cells. Well, litmus milk test involves an oxidation reduction activity or activities. We also know that it is reduced, and you would also have to have an oxidation. They come hand in hand. So if something is reduced, then something in the reaction has to be oxidized. And we see that there was a reaction because the litmus turned blue, and blue represents or indicates an alkaline solution. Oops, spelled that wrong. And FYI, for if you see pink, that means that it's acidic. So here we have a change in the molecules, indicating that there is a blue color change. We see that the litmus milk is reduced. That's reduced. We have oxidation that occurs when we see the results of blue, the litmus being, or the milk being blue, which gives us alkalinity. So our answer is B, because we see oxidized molecules. Which of the following is most likely to be bactericidal? Membrane filtration, ionizing radiation, lyophilization, or freezing or freeze drying, deep freezing, or all of the above? Well, filtration doesn't really kill bacteria. Ionizing radiation, yeah. Lyophilization or freeze drying, well, it'll freeze it, but will it actually kill it? No. Deep freezing, again, it will freeze it, but it will not kill it. It will make it dormant, but won't kill. And it's not all of the above. So what we're left with is B, ionizing radiation. It's ionizing radiation because it can ionize water, which will give hydroxyl radicals. And these radicals can kill bacteria by reacting to organic, by reacting to organic cellular components like DNA. And DNA can be damaged. So the answer is B.